are now groveling. It's my pleasure to welcome Dr. Brad Hill, who is a professor in the Department of Medicine, as our speaker today. He's also the Associate Director of the Center for Cardiometabolic Science, which you can see. Dr. Hill is a graduate of Trevecca University in Nashville and then did a master's at Fisk University and completed a Ph.D. in biochemistry and molecular biology with Aruni Bhatnagar here at UofL. He subsequently did a postdoctoral fellowship at the University of Alabama in Birmingham, and it's there he pursued his research interest in free radicals and mitochondrial biology and metabolism. He was recruited back to UofL in 2009, and he's been very successful in research. To wit, he's already a professor. And he, in 2028, it's not on that slide, it was on the notification, he was elected a fellow in the American Heart Association. That's the FAHA, which was in the seminar notice. He's a highly regarded and very generous collaborator, including with me. He's trained Ph.D. students, 3 of them. He currently has 4 Ph.D. students working in his lab, and he's also trained 6 postdoctoral fellows. And he has served as a mentor for a number of undergraduate students and faculty members in his department. He served on review panels and is a reviewer for a variety of journals and has been awarded for his editorial excellence. And he's an editorial board member for 7 journals, including Circulation Research and the Journal of Molecular and Cellular Cardiology. To date, in 2022, he's published 13 peer-review articles, which again indicates his productivity. He's PI on 3 R01 research grants and has projects on 2 NIH program project grants as well, and is a co-investigator on 2 additional NIH grants. So Dr. Hill keeps very busy, and so he's going to talk about some of his research today, and thank you for talking with us today.
Thank you very much, Carrie, and thank you for the opportunity to present here today. I know this probably isn't a cardiac-centered group, but I did want to discuss some of the work that we're doing relating with metabolism and how it can influence different facets of cardiac remodeling. And some of the pathways I'm going to discuss have also been shown to be active in islet cells, so it could be some crossover there, if you will. So what I'm going to touch on today is really all of these domains of interest that I have. We're really interested in how metabolism influences heart failure, as well as how it influences, with the heart failure, we're going to talk mainly today about fibroblast biology and how it influences fibrosis. We're interested in mechanisms of myocardial development and repair. So the holy grail of that field is to try to make myocytes divide, which is, the heart is a very obstinate organ, does not want to allow its parenchyma cells to divide once you reach adulthood. And we're going to talk a little bit about this during development as well. And then I'm going to touch upon some of the work we've done with cardiac remodeling in response to exercise and how metabolism influences those processes. But really all of these, everything I want to talk about centers around glucose metabolism predominantly. We know the heart, glucose is very important in the heart for its energy production. On the left, you'll see a schematic showing what the heart uses for ATP generation. It primarily uses fatty acids and secondarily uses glucose to make up most of its ATP. But it's a very flexible organ. It can use lots of different substrates. It can use lactate, for example, during exercise. Lactate levels go up quite a bit in the bloodstream, and it can use lactate quite avidly. It can also use ketone bodies. And some of the other substrates it can use but doesn't very much are BCAAs and glutamine. It really doesn't use very much of those. Pyruvate, which we commonly use in our cell culture mediums at 2 millimolar concentrations in circulation, that's less than 150 micromolar. So it's not really a significant source of ATP for the heart endogenously. And it doesn't really use acetate unless you've been drinking a whole lot. If you drink a lot of alcohol, acetate levels rise to millimolar levels, and the heart will then use it for energy. But the interesting thing about glucose is that although the heart takes it up and can use it for an energy source, the fate of glucose is really quite unclear, and it's been shown that more than 50% of glucose that enters the cardiac myocyte is not fated for catabolism. This has been shown in some studies in humans in the 80s. There are very nice studies by Judith Wisniewski and Edward Gertz and colleagues, and this did some work during exercise as well. And we've done this in rodent cardiomyocytes as well. So where does the glucose-derived carbon go? Well, glycolysis is a very interesting pathway, even though it's one of the oldest pathways that we know of. It's got many of these what we call amphibolites. These are metabolites that can be used either for the carbon can be diverted down the glycolytic pathway to make ATP, it's substrate level phosphorylation, or these can be diverted into ancillary biosynthetic pathways, such as the pentose phosphate pathway, the hexylstream biosynthetic pathway. It can provide carbons for the glycerol backbone of glycerol phospholipids, and it can provide carbon for the serine biosynthesis pathway that makes both serine and glycine, as well as it's important in one-carbon metabolism. So, and even within these ancillary pathways, there's branch points that are further like a secondary amphibolite downstream. So it's what I'm really interested in is how the heart coordinates all of these processes to maintain heart size, to grow if it needs to grow, and then how this could go awry in the context of pathology. So really this is, if we wanted to drill down and say what is the real single question that we're addressing, is really how do cells coordinate substrate partitioning to meet the demands for growth and function. And what I have on the left here is just kind of a very simple schematic of a substrate going into a cell, and from the substrate you can generate metabolites that create the building blocks for example, the cell membrane, for nucleic acids and amino acids for proteins. And then these turn over as well. So the substrate, as I mentioned, is used for growth and repair. These would be more anabolic pathways, but it's also used to maintain our body heat, to maintain our temperature, and for basic movement. And these are the catabolic pathways that are required for ATP generation. So what I'm going to be addressing today is really how metabolism is coordinated to actualize, form, influence function of the heart, and how it can become discoordinated in the context of disease. And what I want to start with is some of the work we've done in the context of exercise and looking at cardiac growth that occurs, like, for example, in endurance athletes. And then I'll transition to study more of what happens in the context of pathology, such as myocardial infarction, and discuss a little bit about what the fibroblasts are doing and how metabolism or changes in metabolism there can influence the extracellular matrix production that is prominent in diseased hearts. And then at the end, I'll also touch on interesting new insights that we've developed on how metabolism could influence myocyte division, which really is what we hope to achieve in the future. So I wanted to start with a study we published in 2017. I'm not going to go through a lot of this, but just kind of give you the highlights of this. I had a student in the lab, Andrew Gibb, who's now at Temple University, and he was interested in exercise. He came to the lab and said, I want to do something with exercise and the heart. I said, let's do it. And so one of the things we wanted to know is what's really happening with glycolysis during exercise. It's been a very difficult question to address. And to really cut to the chase, what we found is that during the exercise bout, what we found is that at the level of PFK, which is it's a committed step in glycolysis, that what we see is a decrease in PFK activity. And this is really regulated by several things, but one of the effectors of this is fructose 2,6-bisphosphate, an allosteric effector that activates the Pkm2 enzyme and antagonizes all the negative allosteric effectors that can occur in the context of the cell. And what we found, we used an antibody against this phosphorylation site of Pkm2 that promotes its activation to promote this fructose 2,6-bisphosphate generation. And what we found was pretty profound, is that a very stark decrease. This is one bout of exercise of mice on a treadmill, and then immediately after, when they come off the treadmill, we euthanize the mice and excise the heart and measure phospho-PFK2 by Western blotting. You can see that this remarkable decrease in phosphorylation of PFK2, and it's well known, this is just only one index of glycolysis. It's very hard to measure this in vivo at the time, so we have to kind of use other processes to try to understand this a little better. And it's well known that PFK, if you have a decrease in PFK activity, a lot of the carbon will be diverted to glycogen. And that's exactly what we see. We see an accumulation of glycogen in the heart muscle right after the exercise bout. So this is kind of opposite what you see in striated muscle, skeletal muscle, right? And this has been a very reproducible finding. It's always nice in the lab when students can reproduce the previous students' findings. And this is a recent student, Kyle Fulgham, was able to reproduce this quite well and see that, again, the decrease in the phosphorylation of PFK2. And he went a little bit further and looked at, the upstream kinase that phosphorylates PFK2, and AKT is also down. And so we're still trying to figure out why this occurs. But we also see that protein phosphatase activities are elevated as well. So for some reason, the heart is decreasing glycolysis, and we wanted to see what the effect of this decreased glycolysis was. So what we did is first, I'm showing this glycogen level again here in wild-type mice. If we exercise these mice acutely, you see an increase in glycogen. If you exercise them for 4 weeks, this is 5 days a week of exercise on a treadmill, a relatively strenuous exercise regimen, we can see an increase in cardiac size as the heart weight to tibia length. And you see about a 10 to 20% increase in heart rate, which is what happens in humans that are endurance trained. We see an increase in capillary to myocyte ratio, so there's an increase in angiogenesis in the heart, an increase in capillary density. And we see an activation of what's known now as the exercise gene program. And this has been shown to decrease in CP beta, and this takes the brakes off of SIDED4 and allows it to be expressed. And really, this increase in SIDED4 has been shown to be required for exercise-induced cardiac growth. So it's very difficult to understand how PFK is contributing to this, but when we got this result, we talked with Paul Epstein, who used to be at the university, and he had made what we call glyco-low mice, where he had driving a kinase-deficient Pkm2 construct. Again, this is the enzyme that can phosphorylate Pkm1, or that can increase fructose 6 bisphosphate to activate Pkm1, and he mutated the kinase domain so that it would be a kinase-dead mutant, but it would have the phosphatase activity. So it actually uses fructose 2,6 bisphosphate and decreases the allosteric effector. And if we just excise these hearts and perfuse them with radial-level glucose to measure radial-level water as an index to glycolysis, we see what we would expect, a decrease in glycolysis, the glyco-low hearts. And we also have another mouse we call glyco-high, which is kind of the opposite of this mouse, where the phosphatase domain is mutated, where it's phosphatase dead and it's kinase active. And in these mice, you see an increase in glycolysis. So it works quite well. And as expected, the glycogen levels go up in the glyco-low hearts and go down in the glyco-high hearts. And if we just take sedentary glyco-low mice and just examine their hearts, what you can see is an increase in cardiac size. It's very similar to what we see in exercise-induced cardiac growth. An increase in capillary myocyte ratio that kind of phenocopies what we see with exercise in wild-type mice, and without any exercise and activation of this gene program. So there's something about this decrease in glycolysis that's triggering this program that's contributing to this physiologic hypertrophy of the heart. So when a new student, Kyle Fulgen, came in the lab, we wanted to go in this a little deeper, and what we realized, we really don't understand what are the real metabolic changes happening with exercise. We know that the glycolytic activity is decreased, but beyond that, we really didn't know much. So what he did is first developed a model. We wanted to know when the heart was hypertrophying. So we want to know the time course of hypertrophy after exercise. So what he did is, these are in male mice, and he had groups where he acutely exercised them. This is 1 day of exercise and measured the heart size. He allowed the mice to exercise for 1 week, and you can see they're starting to increase, but it's not quite significant. And by 2 weeks, we have exercise-induced cardiac growth that doesn't go any further after that. So we know that this is the active phase of growth at 1 week. And so what he did is he standardized a method of C13 labeling in vivo using a non-invasive delivery of C13 glucose through a liquid diet. And so in this, we feed these mice the liquid diet for 18 hours after the exercise bout, and then measure what happens, where we see the C13 showing up in the metabolite pools in the heart. And I'm showing mainly male mice here because we found this interesting finding. In female mice, they exercise just as much, but we don't see any cardiac growth in female FEV mice. So for the rest of this part of the talk, I'll just talk about male mice, but we can talk about the female mice a little bit later if you'd like. So in this study, what Kyle did is he first acclimated these mice to the liquid diet. So this is just the liquid diet that we feed them with normal glucose in it. In the final 18 hours before euthanasia, we switched the diet to a C13 glucose-labeled diet. So all the carbohydrate in this diet is C13 glucose. And he performed the different exercise capacity tests, and he showed after 1 week the mice are already showing an increased ability to do work that kind of maxes out by 4 weeks. Their distance run is farther. And one of the critical facets of this approach is if the mice are consuming more of the diet, then it's hard to interpret the data, right? But we found that they consume the same amount of diet over the course of this feeding. It doesn't matter whether they exercise for 1 day, 1 week, or 4 weeks. And the body weights were no different at SAC. There was a lot of data from this. We have probably 200 metabolites in this, so I'm not going to go through all of that because I think you'd probably fall asleep because it's too much to consume at one time. But what we saw is that at 1 day of exercise, we start seeing an increase in C13 enrichment in the citrate pools, isocitrate pools, alpha-ketoglutarate, succinate, fumarate, malate. So all these TCA cycle metabolites show an increase in C13 enrichment that tends to go back toward baseline or sedentary levels by 4 weeks. So in this active phase of growth, at 1 day and 1 week, we see an increase in C13 labeling. And we've looked at the total metabolite abundance. So this is not C13 labeling, just the total abundance of each of these metabolites. We found something quite interesting is that at 1 day, we see a decrease in alpha-ketoglutarate and citrate quite predominantly. And this, to me, screams epigenetic modification. Something's going on with the epigenetic landscape at some level. We haven't gotten into that. But what this suggests overall is that there's an increase in C13 enrichment, but a decrease in abundance, suggesting an increased turnover and keeps utilization of these TCA cycle metabolites. And some of these metabolites through cataplerotic processes can provide precursors for biosynthesis. For example, it can make aspartate from oxaloacetate and glutamate from alpha-ketoglutarate. And Kyle tried to find a different way of presenting this so it's a little bit easier to see in one graph, and we call these Fulgham plots. So this is his last name. But this is just a heat map of the isotopolog. So this is the number of carbons in, for example, glutamate that's labeled with C13. So you either have 0, meaning no carbons were labeled, or 1 carbon, 2 carbons, 3 carbons, 4 carbons, or 5 carbons in the pool. You can see at 1 day and 1 week, there's an increase in the C13 labeling, as well as a decrease in these amino acid pools, suggesting there's increased utilization, increased C13 enrichment from glucose into the TCA cycle pool and into these amino acids, and an increase in turnover. And this is important because these are used in anabolic pathways, for example, of nucleotide biosynthesis, for example. And it's been shown that that cardiac hypertrophy occurs in part because glucose is directed toward aspartate biosynthesis. Another thing that we can get out of these data is absolute abundance of adenylate nucleotides. So from this, we can measure the energy charge that's occurring. This is usually very, very strictly controlled in the heart. You usually see it's like 0.73 or so. And that's what we see in sedentary mice, but we actually saw an increase in the energy charge at 1 day and 1 week that goes back to baseline at 4 weeks. And this is important because for there to be anabolic pathway activity, you want there to be, plenty of energy for the cell. And so this all together suggests that there's an anabolic environment for the heart to grow. So previous studies have looked at skeletal muscle and look what happens early in an exercise training regimen. And you see really remarkable changes in transcripts really early after acute exercise, that once there's adaptation, there's just a more general increase in the transcripts. And same thing with protein content and performance. So we wanted to kind of do this in a way for metabolites. So what we did is did 1 session of high-intensity exercise with FEV male and female mice, freeze clamp the hearts in situ immediately after the bout, 1 hour after the exercise bout, or 24 hours after the exercise bout, and then perform some untargeted metabolomics. And this is a heat map of this, and there's a lot of things changing here, but I want to draw your attention to the branched-chain amino acids, because these go up quite a bit 1 hour following exercise. Each one of them do. And they also go up in the plasma as well. So acutely, they're even going up. So this is thought to be due to liberation from skeletal muscle, for example. And this is interesting because BCAAs are known to promote protein synthesis. This is just a schematic out of a paper that shows that the branched-chain amino acid leucine can activate mTOR, which can drive protein synthesis. It's been shown in several studies, just showing an example here, that the branched-chain keto acids that derive from branched-chain amino acid oxidation also can promote protein synthesis. This is called a sunset assay, where puromycin is incorporated into the proteins being translated, and you can measure this by Western blotting. And you also see an increase of in the phosphorylation of 4E-BP1, suggesting that they promote protein synthesis. So we wanted to see, are these BCAAs that are going up with exercise, are they contributing anything to the cardiac growth? So we worked with Martin Young at UAB because he was doing some BCA work as well, and he worked with the company to get different diets made with different levels of BCAAs. So we had a low and a high in our normal child diet as well. The low BCA diet has half the amount of BCAAs that a normal child does, and the high BCA diet has double the amount. And what we found is, although the high BCA diet didn't show a further increase in circulating BCAAs, the low BCA diet shows about half the level. So then what we decided to do is to put the mice on this diet and exercise them for 2 weeks when we know that in which the time frame we know that you get maximum cardiac growth, and see what this BCA diet does. One of the concerns going into this was that the mice getting a low BCA diet might not exercise as well, which would obfuscate our results. But we found that it doesn't. The mice still increase their distance run and their exercise capacity after the 2 weeks of exercise. And what was interesting in this study is that although the high BCA diet we saw at exercise-induced cardiac growth, we did not see exercise-induced cardiac growth with the low BCA diet. So this suggests that this BCA burst that happens after exercise may facilitate cardiac growth. So collectively for this part of the talk, what we think is happening is there's this coordinated response in metabolism that helps coordinate growth, in which BCAAs are liberated in the bloodstream. We don't know if that has a role in the decrease in PFK. We don't know that yet, but we do see that the energy charge increases and the glucose-derived carbon is diverted to amino acid synthesis, which is important for growth as well. And we think this all works together to promote cardiac growth. So we're still working on some of these studies. And we hope to have more to report at some other time. What I'd like to do now is switch to some of the work we're doing where we're looking at metabolism in the pathological context, such as what occurs in myocardial infarction and pressure overload. So both of these, you also get hypertrophy of the heart like you do in exercise. But what's common in a stressed heart like this, a pathologically stressed heart, is you can see an increase in fibrosis. And with MI, you see a lot of extracellular matrix production for this replacement fibrosis, and you also get some fibrosis in the non-infarcted regions as well. And when we started thinking about this, I wanted to know how much ECM can fibroblasts synthesize. So I was looking through the literature to see if anybody had quantified this, and actually in the 80s, there was a very interesting study where they took fibroblasts and they were trying to just quantify how much procollagen is made per fibroblast. And all of these different indicators here are just different passages of the fibroblast, but what they found is that a single fibroblast can make 500,000 procollagen chains per hour. It's an enormous amount of collagen that the cells can make. And trying to find other ECM factors such as hyaluronan, which is also made from glucose. In the 60s, it was published that a fibroblast cell line 3T6 cells synthesize hyaluronan at roughly 15 times the rate at which they synthesize collagen. So that's an enormous amount of collagen that these fibroblasts have to make. So this suggests that metabolism has to remodel quite a bit to make all of this extracellular matrix, at least in the context of myocardial infarction. So the question we have then is, how do fibroblasts sustain their capacity to produce ECM? And there was a nice paper by Rachel Chambers' group a few years ago that showed that glucose is absolutely required for this, and that uses glucose for de novo glycine synthesis. And this is some proof of principle data showed here, where they treat fibroblasts with TGF-beta, which is known to activate fibroblasts and make them make a lot of extracellular matrix. If you have glucose in the medium, they do make a lot more collagen. But if you leave glucose out, they're not able to increase the collagen with TGF-beta. And so we're very interested in trying to understand this process and understand how glucose is being diverted to glycine to make collagen. We focus on glycine because glycine is the predominant amino acid in collagen. This is just a pie chart showing the different amino acids that are in the collagen protein. The majority is glycine and proline can come from glutamine as well. And we have Andrew Gibb, who was a student with me at Temple. He kept on going with some fibroblast work with John Elrod, and found that glutaminolysis is essential for myofibroblastic persistence, and you could target it to reverse heart failure as well. So all of this suggests that there's really a vast need of metabolism to to meet the material demands for ECM synthesis, and there must be some sort of amplification mechanism in place. And when looking at this, it's hard to figure out where that could be, but one way it could occur is by driving carbon from the Krebs cycle back up into the glycolytic pathway. And if it drives it up into these regions, that's important because these are reversible reactions in glycolysis, and so it could be driving more carbon into glycine to make collagen. So this brings us to the metabolic cycles that was in the title of my talk. So there are many metabolic cycles that are known in biology. The glucose cycle is not really well known in the liver. It's important in gluconeogenesis, and it's the glucose-6-phosphatase enzyme that's important to make glucose from glucose-6-phosphate. In bumblebees, there's a really well-known fructose-6-phosphate cycle that uses cycling between PFK and FBP to promote a futile cycle to make heat. So when the bumblebee needs to fly in cold weather, it'll start activating the cycle to make heat. And then there's a phospholipid pyruvate cycle, which is a little bit more complicated cycle because it involves more than two enzymes that converts PEP to pyruvate, but then pyruvate can enter into the TCA cycle, and also other carbon sources can contribute to this. And then phospholipid pyruvate carboxykinase can convert oxaloacetate in the TCA cycle back to PEP, which would take it right back up into that lower portion of glycolysis. So all of these cycles involve gluconeogenic enzymes, and there's really nothing known about gluconeogenic enzymes in the heart, and you wouldn't think that the heart would have gluconeogenic enzymes, right? The hypothesized purpose of these cycles are energy dissipation, of course, thermogenesis, as shown for the bumblebee, and control of metabolic intermediates and biosynthesis. So we started wanting to look at this in the context of a diseased heart, specifically in myocardial infarction. And I'm showing this schematic here showing these cycles in different form. This is the glucose-6-phosphate cycle, the fructose-6-phosphate cycle, the PEP cycle. And you can see that it's all integrated quite well. And you can see that hyaluronan, I mentioned that as an extracellular matrix protein, it's made from glucose as well. And that glycine is made from glucose. So there's a lot of draw of glucose-derived carbon to make these things. And you would think there would need to be more carbon. This could be one way in which you could drive carbon, the amplification mechanism to drive carbon from the TCA cycle back up to PEP so that it can get into the glycine biosynthesis pathway. So what we wanted to do is first look, does the heart express any of these gluconeogenic enzymes? So we isolated adult cardiomyocytes, adult cardiac fibroblasts, cardiac coronary endothelial cells, or aortic smooth muscle cells, a model of smooth muscle cells. And these are just the markers showing that these are the cells that we believe that they are. Endothelial cells express enos, cardiomyocytes express troponin, cardiac fibroblasts express quite a bit of vimentin, and both cardiac fibroblasts and smooth muscle cells express alpha-synosyl actin, depending on the activation state. And then we started looking at the different gluconeogenic enzymes. I'm not showing all of them here, but the ones that I think are very important. The heart doesn't express any PCK1. That's mainly expressed in the liver. But the heart does express PCK2, and predominantly in the non-myocytes. So in cardiac fibroblasts express really high levels of PCK2, and endothelial cells and smooth muscle cells also express it. Interestingly, the heart expresses very high levels of fructose-bis-phosphatase 2. So the myocytes don't express the first enzyme in gluconeogenesis, but they express these other two. That's something we don't know what those enzymes are doing there. And then we're also interested in the pyruvate kinase because it's a part of this PEP cycle. And the myocytes only express Pkm1 isoform. They don't express Pkm2, at least in the adult heart. The cardiac fibroblasts predominantly express Pkm2. So the fibroblasts seem to have an intact PEP cycle that could be used to drive carbon into glycine. So the next thing we wanted to do, I'm just showing this again to keep your mind on this. We wanted to know what happens in the context of myocardial infarction. So we infarcted mice. This is permanent coronary ligation. And then we had a sham group as well. And we looked at just the expression of PCK2 and Pkm2 in whole heart homogenates from sham and MI hearts. And what you can see is a pretty robust increase in both of these enzymes. What we also did is isolate fibroblasts from the infarcted heart, or took naive fibroblasts and treated them with TGF-beta to activate them and look at the expression of these enzymes. And we did some RNA-seq on this, and from this we could readily see that the fibroblasts from infarcted hearts have a robust upregulation of PCK2. And if you just took naive fibroblasts, and we usually treat these with FGF to keep them in the inactive state as much as possible, or TGF-beta to activate them, we see that this also increases PCK2 expression. Pkm2, it's one of the splice variants of pyruvate kinase. It also goes up in MI. This is a Western blot showing Pkm2 in fibroblasts isolated from the infarcted heart. I'm showing alpha-muscle actin because this is an indicator of their activation state. As expected, they'd be activated in an infarcted heart. And if we take naive fibroblasts and just do PCR after treatment with FGF or TGF-beta, and the SS is just serum-starved fibroblasts as an additional control, we can see that Pkm2 expression goes up as well. So this suggests that this PEP cycle is in the fibroblast and it's upregulated in the context of myocardial infarction. We also did some imaging, and this is images of sham and infarcted hearts. And this is alpha-sarcomeric actin to identify the myocytes. You can see that they're a nice green color. And Pkm2 is really not expressed in the myocytes at all, as shown by our immunoblotting results. But Pkm2 seems to be expressed in the interstitium where the fibroblasts reside. In sham hearts, in the infarcted heart, you can start seeing in the border region next to the infarct, seeing these really big swaths of Pkm2 expressing cells, which are most likely fibroblasts. So the next question we ask is, might these Pkm2 splice variants influence collagen production in post-MI remodeling? And so there's a lot of biology behind Pkm2 that's been worked out mainly in the context of cancer, because cancer cells express really high levels of Pkm2. And Pkm2 can be in both a dimer form and a tetramer form. And Pkm1 is only in the tetramer form, and it has a very high catalytic activity. So if you focus your attention here, if you have a Pkm1 expressing cell, you'll have higher activity, and this would drive more phospholipid pyruvate down into the TCA cycle and promote kind of a feudal cycle, if you will, because it would pull a lot of these carbons down. And then Pkm2, it's in both of these forms. If it's in the tetramer form, it phenocopies Pkm1. It's a dimer form. It has very low activity, pyruvate kinase activity. And so we bought a mouse from Jackson Labs, a mouse that Matthew Vanderheiden made years ago. And these mice have LOXP sites surrounding exon 10 of the Pkm allele. And so if this is floxed out, then what you end up with is a Pkm1 splice variant. And so we wanted to first validate these mice work the way we thought they did. So these mice were bred to a fibroblast-specific Cree driver. And then we treated with tamoxifen for 5 days and then gave a 5-day washout period, then isolated the fibroblasts and looked at Pkm2 and Pkm1. You can see Pkm2 is starting to go down and Pkm1 goes up. So then they asked the question, what happens with MI in this context? So we, again, treated these mice with tamoxifen for 5 days, had a washout period, then performed a baseline echo, and then performed permanent coronary ligation, and 4 weeks after, examined the hearts by echocardiography, and then had tissue harvest to look at other biochemical endpoints. And these are the data. What we actually saw was a decrease in diastolic volume and in systolic volumes. It's suggesting less dilation of the ventricle. We saw an increase in ejection fraction in the hearts in which the fibroblasts expressed Pkm1. There's an increase in the wall thickness, so there's less wall thinning whenever the splice variant is switched from Pkm2 to Pkm1, and the volumes of the ventricles, the diameter of the ventricles is decreased, suggesting less dilation overall. And you can also take the lungs out and look for congestive heart failure, an index of congestive heart failure, and we see a decreased wet-dry lung weight. In a subgroup of these mice, we took for hydroxyproline measurements to measure collagen, and collagen levels were down as well. So it suggests that this splice variant switching is an intervention that could potentially improve outcomes after decreased fibrosis after myocardial infarction. So how can we actually interrogate this a bit more? What I'm showing here is a little bit modified schematic. Everything that's bolded was from our RNA-seq results. These are all the enzymes of the glycine synthesis pathway. These are all upregulated in MI fibroblasts. PFK1 and PFKP3 were actually upregulated as well, and Pkm2 and PCK2 were upregulated. And what we think is happening is that Pkm2 is upregulated to actuate a PEP slow cycle that will decrease the conversion of this carbon into pyruvate, leaving more available for going the glycine pathway. And we think that there's also this additional amplification step driving carbon from PCA cycle up to PEP to drive more into glycine. So how can we interrogate this cycle? Well, we've kind of accumulated several tools to look at this. We can make a PEP slower cycle, if you will, by converting all the Pkm1 that's there into Pkm2, if you will, to make a more anabolic cycle. The mice that I used previously, we call these the PEP fast form, in which you convert Pkm2 to Pkm1, and we see that this, as I showed, improves outcomes after myocardial infarction. And we've now have mice where we can delete the PCK2 arm, where we don't have that return of carbon back up into the pathway, called a PEP no cycle. So we're currently in studies trying to understand really in detail how each of these interventions affect remodeling after myocardial infarction. And there's some therapeutic possibilities with this as well. PCK2 inhibition, there's not many options right now. There's 3-mercaptopicillinic acid, which is relatively nonspecific. We're going to try it and see if it works. But there are some really nice Pkm2 activators. And when I say Pkm2 activators, this promotes the tetramerization of Pkm2. It takes it from the dimer form to the tetraform so it can mimic the Pkm1-like activity. And there are several to choose from that are commercially available or that are are being made right now and tested. So what we did is we, as I mentioned, if you convert this to the dimer, if you have a Pkm2 in the dimer form, you'll have a decrease in pyruvate kinase activity, which would drive more carbon for collagen synthesis. And if you could have an activator that promotes the tetramerization of Pkm2, this would activate this enzyme and decrease glycine. So we wanted to see if we can see this tetramer dimer form in the cell, in the fibroblast. So it was a postdoc that worked with me a few years ago, Pyromera, that worked out the use of this cross-linker, disuccinyl superate, it's hard to say. But she first just did a dose response to see what she didn't want to get over cross-link the protein, and that's what happens when you use a lot of it. But she found that 0.5 millimolar works really well to see the tetramer form. And we treated these cells with the ML265 Pkm2 activator, and we can see we can increase the tetramer form. And if we do this and then treat the fibroblasts with TGF-beta and do a dose response of the Pkm2 activator, we see a dose-dependent decrease in collagen production in the fibroblasts. And this is there's another proprietary compound we got from Pfizer that it's in development, and it does the same thing. So this suggests that this is some sort of actionable therapy that could be used to decrease fibrosis in the heart. And there's really no current therapies that address fibrosis in the heart. So this is kind of our working model. The PEP cycle, we think that fibroblasts that are in the infarcted heart kind of use the PEP slow cycle to drive carbon from glucose and glutamine into glycine to make more collagen. What we see is if we use the PEP fast mice, where we can promote the splice variant switch to Pkm1, this drives more of the carbon into the TCA cycle pool, leaving less available to make collagen. And we're still in the process of testing the PEP no cycle, which is a student lab. Colin Wells is testing this part of it. And what we expect is we'll see a decrease in collagen as well, because the glutamine-derived carbons can't contribute to collagen in that case. So I've discussed these metabolic cycles, and I think they're very interesting because they may have many other functions. And I've shown that that the myocytes express glucose-6-phosphatase, they express FPP2, so these cycles may be active in the myocyte. And in the PCK2, it can also be expressed in the myocyte only during development. And we see that Pkm2 is expressed as well as PCK2 in the fibroblast. And what's interesting is going back to those glycolytic mice I discussed at the beginning, we wanted to see, well, is FPP2 responding to this decrease or increase in glycolytic activity that happens at the level of PFK? And whenever you have a decrease in PFK activity, what we see is almost looks like a knockout of FPP2. It goes down. But if you activate PFK1, we see this going quite up. So it is something that the heart is trying to really balance its metabolite pools to make sure that it can still continue to function. And some of the work we've done, we've started looking at what happens in adult myocytes versus neonatal myocytes. Neonatal myocytes at day 1 after birth proliferate avidly. And we see it in adult myocytes don't express PCK2, but neonatal myocytes do. So we think there may be some role for this in chondromyocyte proliferation. In addition, there's a publication in 2020 that showed that Pkm2 also it's really highly expressed in cardiomyocytes of the embryonic and the early neonatal heart, but by P7, whenever the hearts become non-mitotic, it's really going down quite a bit. In adulthood, it's barely visible in the myocyte. what we think is this PEP cycle may also be involved in the switch between the proliferative and hypertrophic forms of cardiac growth. So we started trying to test some of this and try to understand how this could influence myocyte division and biosynthetic pathway activity. And biosynthetic pathway activity would have to be high if you're going to make 2 myocytes from 1. You have to make more phospholipids for the membrane, more nucleic acids for gene transcription and what have you. So we worked with Riham Abelousa and Tamar Muhammad in the Division of Cardiology, and he really published a very nice paper. This paper that I'm showing this schematic is from Cell in 2018, in which he showed that if you overexpress 4 cell cycle factors, CDK1, CDK4, cyclin B, and cyclin D, you can increase myocyte proliferation in vivo in the heart. And he came up with this nice assay using iPSC-derived cardiomyocytes in a dish, which if you keep them for 60 days in the dish, they stop proliferating completely. Then you can express these 4 factors, and you can see an increase in G2M stage of the cell cycle, and an increase in DNA synthesis as well. So we wanted to see if biosynthetic pathway activity might be increased in these. So we first did RNA-seq. And what we found is many of the Krebs cycle enzymes are downregulated in proliferating myocytes. Those myocytes are treated with the 4 factors. Amino acid oxidation, fatty acid oxidation is down, but we start seeing an increase in many of these biosynthetic pathways, such as NAD and ADPH synthesis, HBP, the hexyl-2 biosynthetic pathways, and these enzymes are up in serine biosynthesis pathway. This is the enzyme, the other anabolic pathway I've talked about a lot today. So we wanted to look at what happens to catabolic metabolism. So we took these myocytes, iPSC-derived myocytes, and treated them with the 4 factors or a LAC-Z control. And we can see that after 48 hours, there's a decrease in oxygen consumption rate. So they're decreasing their oxygen consumption, mitochondrial oxidative phosphorylation. We also used another technology that's recently been developed called Recipher, in which you can measure oxygen consumption over a course of several days. And this is where we treated with this 4F virus or the LAC-Z, and you can see that as the virus starts to express, you can start seeing a decrease in oxygen consumption, which validates the seahorse assays shown here. Then we use C13 labeling, stabilized resolved metabolomics, to try to understand what's happening in the biosynthetic pathways. And I'm not going to show all of these data, it's quite a bit, but you can see, for example, NAD, C13 incorporation in NAD is much increased. So this is the N+0 isotope log. This is the NAD pool that doesn't have any C13. You can see that pool with F4F, there's a decrease in the C12 labeled carbon and an increase in the C13 labeled carbon. And this is at the N+5, meaning there's 5 carbons labeled. And this comes from the ribose ring to make NAD. And we see similar things in the other ancillary biosynthetic pathways as well. This is UDP hexNAC, which is used for several things, including to put on and off of proteins for protein glycolation. And we see a similar phenomenon here, where an increase in C13 labeling in this really complex molecule. So we went on this paper to then inhibit a step in each one of these pathways, and it didn't really matter which pathway we inhibited. If we inhibited any of these pathways, we completely blocked proliferation. So what we think is that these pathways are very important and critical for myocyte division. If we inhibit it, we inhibit myocyte division. And then, so this was all iPSC, so we wanted to look back into a more physiologic scenario, not just ectopic expression of these 4 factors. So we again went back to the adult myocytes and neonatal myocytes, and looked at the transcript for the enzymes in many of these ancillary biosynthetic pathways: polyol pathway, the pentose phosphate pathway, glycogen synthase pathway, nucleotide biosynthesis, NAD synthesis, glycerolipid, hexosamine, serine. I think you can appreciate that there's a much higher transcript level for all of these in the proliferative myocytes. So suggesting that proliferative myocytes have to have higher biosynthetic activity. We wanted to see if this actually happens in vivo. We went back to this in vivo labeling approach. So this is work with Helen Collins. She was interested in what happens in the pregnant heart. She's interested in the mechanisms of maternal cardiac growth during pregnancy. But we could use this as well to look at the pups. She was doing her studies and at the same time was isolating hearts for us from embryos at day 16 of pregnancy. And then we also got the pup hearts from P7. The mice received diet through the mother, of course, and then did stabilized hope resolved metabolomics. And I'm just showing kind of a snapshot of the results here. This is all of these metabolites are derived from 6-carbon glycolytic intermediates. And you see that all of them aren't elevated. Sorbitol looks exactly the same. And E16 is the non-proliferated myocytes versus the proliferative myocytes of the hearts from the P7 mice. But many of these metabolites in the nucleotide biosynthetic pathway and pentose phosphate pathway showed lower enrichment in the non-mitotic heart, especially NAD. And then from the 3-carbon glycolytic intermediates, we saw a very similar thing. Glycerol 3-phosphate was lower enrichment whenever the hearts became non-mitotic, glycerol, phosphoglycerol, and serine. But this wasn't something that happened across the board because we saw an increase in aconitate C13 labeling, suggesting that carbon is being diverted to glucose oxidation rather than going in these biosynthetic pathways. And this makes sense because the heart's having to work under a harder load and has to generate more ATP to continue contraction. So this is thought it was quite interesting that we could do this in vivo. So I want to return back to this PCK2 module, looking at the PEP cycle. And it's been shown that Pkm2 does promote myocyte proliferation, but it hasn't been looked at for PCK2. So we use this iPSC-derived cardiomyocyte platform to really understand if PCK2 has any influence on myocyte proliferation. And what you can see is these cells do not express any PCK2 at baseline, but if you overexpress it, of course, you can see it there. And we can see the normal response when we give the 4 factors, an increase in cardiomyocyte proliferation. By itself, PCK2 doesn't do much, but it amplifies the response, amplifies proliferation if you transduce the cells of PCK2 as well. So we think this amplification step is probably very important to drive maximal proliferation of cardiomyocytes. What I've kind of shown in these last few slides is that what we think is happening, we think the developing heart has an active PEP cycle, and they have a PEP slow cycle, if you will, because they have Pkm2 and they've expressed PCK2 as well, and we think this amplifies biosynthesis. And the adult heart does not have a PEP cycle in the cardiomyocyte, and this would lead to lower biosynthesis, and we think this is critical for one of the reasons why the adult heart myocytes do not divide after infarction, for example. So I want to kind of stop there. It's a lot of information, and summarize kind of what I've shown today. I showed that exercise promotes metabolic remodeling that integrates both BCA metabolism with glucose metabolism to create this anabolic environment for cardiac growth. I showed that there are several metabolic cycles at play in the heart. The myocytes express glucose 6-phosphatase and fructose bisphosphatase. We don't know what this does yet, but we know that the PEP cycle likely plays an important role in both fibrosis as well as cardiomyocyte proliferation, depending on the cell type that the cycle's active in. And overall, if you take nothing else away, just take away that the metabolic network is very flexible and provides several solutions for providing the building blocks for making extracellular matrix in the context of fibroblasts, or can help the myocyte make more building blocks for hypertrophy or for proliferation. With that, I'll close and acknowledge the people that did the work. The early work was done by Andrew Gibb, who's now at Temple University, and then Kyle Fulgham, who's here just defended a couple weeks ago. He's been a stellar graduate student, has done a lot of the work in standardizing all this in vivo C13 labeling approach. Has published 6 papers this year, so I think he's done very well. Lindsay McNally, who's no longer here, she was a lab manager. She worked a lot in the Pkm2 project. Colin Wells is working on this PEP cycle project as well. Daniel Nguyen is also an MD Ph.D. student lab. He's working on the fibrosis projects. And then we've had a lot of help from Rehan Abuelisa and Tamer Muhammad for all the myocyte proliferation work. Steve Jones and Helen have really helped a lot as well. And really, all the people on here have been integral for some of the work we've done. So with that, I'll close, and hopefully I can address any questions that you have.
It was lovely biochemistry.
Okay, I was worried I was over, but I stopped a little short.
So I have a question. For one thing, and ask about something that's related to endocrinology, pulsatility and growth hormone. I don't know if you saw the paper in Science, maybe by Holly It was looking at different. Turns out it's due to the pulsatility of growth hormone.
Really?
Yeah,
Interesting. It's really hard to tease that out. I mean, there's lots of things that play in the female hearts I'm not going to talk about. It has been shown that female hearts hypertrophy just fine in other strains of mice. But in humans, it's been shown that female hearts don't hypertrophy as much. So it's, there's a little bit of a strain-dependent difference, but the female hearts have a completely different metabolic profile. It's pretty pronounced. If you just did metabolomics, compared them just at baseline, there's a lot of differences. And their response to exercise, they show a marked upregulation of ketone bodies. And so one of the ideas that we have now is ketone bodies is kind of a starvation signal, right? It means you need more energy. So I'm wondering if the ketone bodies are somehow antagonizing this as well. So it may be something like a hormone-driven phenomenon, like you said, or it may be something that's more fundamental in the metabolic chain differences between male and female mice. Well, all of these studies were done. Everything like the mice are always exercising at 9 a.m. We used FEV mice, which are blind at weaning, but they still see light, so they still should have some circadian rhythms. But we haven't looked at differences in circadian. If there's a difference in circadian rhythm between male and female, it could account for that. Martin Young at UAB found he has a really interesting study published in JMCC where he showed that if you just feed a high BCA diet for 4 hours, at the end of the day, the hearts become bigger after that 4 hours. I couldn't believe it at first. It's just 4 hours of this feeding this high BCA and they go bigger. They become bigger. If they feed that same diet early in the day, it doesn't happen. So there's something to do with the circadian biology that maybe there's a certain time of the day where growth and repair and everything like that happens normally. And that could be very important for athletes and things to know when maybe they need to sync themselves up when they can maximize their training and growth potential.
How old are these?
These mice are anywhere between 12 and 20 weeks of age. So they're young mice. They're relatively young mice. We've got a large project going with aging. It's mainly concerning myocardial blood flow in the aged heart, but we're starting to work in exercise there. We do see pretty robust decreases in exercise capacity in aged mice, as you would expect, but we haven't done enough for me to answer that with any specific answer.
Okay. You used ML265 and another drug. Do they have impact?
Well, I mean, they're a drug, they're going to go everywhere, right?
Yeah.
Pkm2, to my knowledge, isn't expressed in the hepatocyte. I think they have PKL, the liver isoform of pyruvate kinase, but there's fibroblasts in the liver, so it could affect other cell types in the liver. My recent grant submission, the critique said, I have this ML265 intervention. They said, well, how are you going to know specificity? Well, it's a drug that's going to go everywhere. So it's a difficult question to get at. But yeah, it's going to go everywhere.
And lastly, with the connection with
we do have data on that, and it does change with proliferation. But we haven't looked at that in any of our models where we change the PEP cycle. It'd be something interesting to look at because we haven't gotten to the epigenetic stuff yet. It's just a little daunting for me. But I think there's a lot there to be uncovered because one carbon metabolism is going to change. Okay, chat. Okay, how do you correlate it to the human heart metabolism, especially in relation to collagen synthesis? Well, it's hard to do these studies in humans, of course, but we can isolate fibroblasts from the adult heart, and they've always been shown to respond very similarly to TGF-beta and things like that. And one of the studies I just mentioned, the study by Andrew Gibb, he did isolate fibroblasts from human hearts and showed that if you inhibit glutaminolysis, you can revert the fibroblasts back to a more quiescent fibroblast state. In the context of metabolism, exercise, we haven't done anything with that as of yet. why you chose 13C and what is its half-life. We chose 13C because it's a non-radioactive isotope, so it's perfectly safe. We all have C13. We have 1.1% of the abundance of our carbon is C13. We have to correct for that in all of our studies. So that's really the best way to trace this in vivo without having a lot of radioactive problems. And then in humans, the collagen comes from diet, not from glycine. Well, you can take collagen in from the diet, of course, but you break it down. You don't just take collagen and redeposit it somewhere. The collagen will be broken down into its amino acid components and then can be utilized. But it has been shown that the de novo synthesis of collagen is required in fibroblasts to promote fibrosis. And those are studies showing that when they leave out glucose, they don't see collagen go up, TGF-beta. And if they give glycine in the media, it doesn't do that much. So it suggests that it's a de novo synthesis that's required. And if so, what are the best sources of collagen? Oh, well, I don't know. There's a lot of supplements out there where you can take collagen peptides.
Aging women.
Yeah,
Every time I go to the
I don't know. That probably would increase the levels of circulating glycine and proline, I would think. But because de novo synthesis is required, I would think it wouldn't make that much of a difference in fibrosis, per se, which is a good thing. If you take collagen, you don't want to be fibrotic. Nice. Any other questions? No? All right. Well, thank you very much. Yes. Tamoxifen?
Is it tamoxifen?
Tamoxifen, yeah. Yeah, it's nasty. I don't like injecting the mice because you put it in corn oil and have to inject them, and we just got quotes for a tamoxifen in the chow, be less invasive and less hard on the mice having to pick them up every day to give it in the diet. So we're going to look towards switching to that. But yeah, we use tamoxifen. I think it's converted to 4-hydroxytamoxifen in the liver, and that's really the active compound that promotes the recombination in our pre-drivers. All right. Thank you.
So much.